I'm Helen Patience, and I'm going to be introducing you to anti-infectives. Anti-infectives. So, what are anti-infectives? Anti-infectives, these are medications or drugs that are used to treat infections caused by microorganisms. These are medicines, stroke drugs that are used to treat infections caused by microorganisms. So, what are microorganisms? Microorganisms are tiny organisms that cannot be seen by our naked eyes. Microorganisms These are tiny organisms that can't be seen by our eyes. What are some of the characteristics of microorganisms? We very well know that characteristics of microorganisms. We very well know that microorganisms are capable of reproducing. They reproduce. They are pathogenic. They can survive in different environmental conditions. They can survive in different environments or environmental condition, and they are diverse. Meaning they can occur in different forms. After discussing the characteristics of microorganisms, we also need to know the classification of microorganisms. Classification of microorganisms. They are grouped into five. First, we have bacteria, we have virus, we have fungi, we have parasites, we have protozoa. Each of these different classifications have different drugs that are capable of treating them. And these drugs are the ones that are known as anti-infectives. For example, for the bacteria, they are treated by antimicrobes, stroke Antibiotics. Antibiotics. So the viruses, they are treated by antivirus or antiretrovirus. It can be also called antivirus or antiretrovirus. Then we also have the antifungus. Antifungus. For the fungi, they are treated by antifungus. Parasites, they are treated by antiparasites. For the protozoa, they are treated by antiprotozoa. Antiprotozoa. For each of these 5 classes, are they 5? 1, 2, 3, 4, 5. For each of these 5 classes, we are going to look at each class step by step. From the first one, which is bacteria, viruses, fungi, protozoa, we shall look at the mechanism of action, we shall look at the drugs and everything. So that is the classification. After the classification, we also need to know the different definitions that are going to guide us in this topic of antihypertensives, of anti-infectives. Different definitions. The first one is going to be a pathogen. A pathogen. So, what is a pathogen? A pathogen is a microorganism that is capable of causing a disease. Pathogen is a microorganism that is able to cause a disease. That is a pathogen. A pathogen is a microorganism that is capable of causing a disease. Then, what is an infection? An infection. An infection, we very well know that an infection is an invasion, something that invades your body, is an invasion and multiplication of microorganisms within your body. Is an invasion and multiplication of a microorganism that causes a disease. An infection is an invasion. A microorganism invades your body, multiplies rapidly, and causes what? And causes a disease. That is an infection. Another definition is bacteria. Bactericidal. These are drugs that kill bacteria. Bacteriostatic. These are drugs that inhibit growth of bacteria. These are drugs that inhibit the growth of what? The growth of bacteria. Another important thing that we're going to look at, we're going to look at resistance. 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 What is resistance? Resistance is the ability of a microorganism to withstand the effects of a drug that were able to kill it initially or inhibit its growth. Resistance is the ability of a microorganism to withstand the effects of a drug that initially inhibited its growth or killed it. This is a very important thing in this topic that we are studying, anti-infectives. So we are going to first look at all the different things that occur in resistance. So we have looked at the definition. Resistance is the ability of a microorganism to withstand the effects of a drug that initially killed and inhibited it. This resistance has different types. So you're going to look at the types of resistance. What are the types of resistance? The types of resistance, we have natural resistance. We have acquired, and we have cross resistance. For the natural resistance, we very well know that in natural resistance, the microorganism itself, the structure, the cell wall, the capsule, and let's say the bacteria, it's capable to withstand the effects of what? Of the drug. Then for acquired resistance, that means that within the microorganisms there is going to be different mutations of this organism in its body. For example, the bacteria within its genes, it mutates so that it blocks the initial binding site of the drug. Then cross resistance, we see that the microorganisms obtain resistance from other not obtaining resistance. In cross resistance, you're going to see that once the bacteria resists a particular drug, it is also going to continuously resist other drugs. That is cross resistance. The next thing we are going to talk about, the mechanism. How do microorganisms resist? How are they capable of resisting resisting the different drugs? The next thing we are going to talk about is how the mechanisms of resistance of the different microorganisms. Mechanisms of resistance of the different mechanism of resistance of microorganisms. Number one, these microorganisms are capable of producing enzymes that are going to destroy the drugs or the anti-infectives. Number one is enzymatic inactivation of the drug. Enzymatic inactivation of drug. Number two is going to be development of efflux plants. Development of efflux pumps. And number three is going to be reduced permeability of the cell membranes. These microorganisms are going to reduce the rate at which substances enter within the cell. So we have reduced cell membrane permeability. This membrane permeability. Then the fourth one is going to be biofilm formation. Biofilm. We need to explain biofilm formation. And the last one is going to be alteration of active site or binding site. So these are the mechanisms by which microorganisms are going to resist. We are going to start with the first one, enzymatic inactivation of the drug. What happens? I'm going to give an example of bacteria. This is our bacteria. This is our what? This is our bacteria. Bacteria is going to produce enzymes. For example, the beta-lactamases. They're going to produce enzymes. This is our enzyme. And this enzyme, whenever the drug or the antibiotic enters into the cell, it is this enzyme, the beta-lactamases, that is going to break down the what? That is going to break down the cell. Then for the development of the efflux pumps, still I'm going to give an example of bacteria. For example, within the bacteria, it is going to develop pumps, different pumps. And whenever the drug bypasses the cell membrane, this is the cell membrane. Whenever the drug bypasses the cell membrane, this cell membrane, and enters into the bacteria, the bacteria is going to continuously pump out the what? The drugs, thus resisting it to perform its actions. The next one is reduced permeability. For reduced permeability, any microorganism is going to reduce or block its channels. These are channels. These channels are the ones that allow substances to enter the cell, are the ones that are responsible for permeability. So substances are going to reduce or close the channels that were initially allowing drugs to enter into the what? Into the cell. Then biofilm formation. Biofilm formation. Here we are going to see that different organisms are going to come together. This is an organism. They're going to come together and form communities. These communities are going to be surrounded by a substance. This is our substance. And this substance is going to be called polymeric substance. It is this substance that is going to protect all control this bacteria and prevent efflux or penetration of the drug into the what? Into the different bacteria. Okay, into the different organisms. Then not specifically bacteria, but different organisms. Then alteration of the active site. For alteration of the active site, here any microorganism, let's say the drug, this is our drug, it was binding on a target site, let's say here, when it's going to bind here. What microorganisms do? They change the structure. They change the structure where the drug was initially binding to reduce its what? Its activities. So that is alteration of the what? Of the active site or binding site. Yeah. So those are some of them. Our next thing that we are going to talk about, I wanted to also introduce a little bit of antibiotics. Remember, we talked about previously all the anti-infectives. We talked about all the anti-infectives. I started by introducing the anti-infectives, where I talked about the classification of the what? Of the microorganisms. I talked of bacteria, fungi, I talked of protozoa, parasites, and I said each of that has its anti-infective. For example, the bacteria is having antibiotics or antimicrobials. So I'm going to introduce a little bit of antimicrobials. Then in my next lecture, I'm going to be talking about the mechanisms of action, how these drugs act, the side effects, the fungi indications, which I will talk about all that. So we're going to have antimicrobials, stroke antibiotics. Very well know that these are drugs. These are drugs that treat infections caused by bacteria. It's going to treat they're going to treat infection caused by bacteria. Those are antibiotics. What is the class? I also want to talk about the classification of these antibiotics. That's all that I'm going to teach in this lecture. Classification of antibiotics. Classification of antibiotics, all the drugs. The first one we are going to look at spectrum of activity. Spectrum of activity, that is the first one. The second one, we are going to look at the effect of the drug on the bacteria. Effect of drug on bacteria. Then we are also going to look at the source of the antibiotics. And lastly, we are going to look at the mechanism of action of the antimicrobials. So this is the classification of the antimicrobials or the antibiotics. So under spectrum of activity, we have the narrow spectrum, and we also have the broad spectrum. For the narrow spectrum, these are going to work on very few microorganisms, but for the broad spectrum, they are going to work on both gram-negative and positive bacteria. Then effect of the drug on the bacteria. Here, on the effect of the drug of the bacteria, first of all, there are some drugs that are bactericidal, and some drugs that are bacteriostatic. For the bacteriostatic, it means they are going to kill the bacteria. Then for bactericidal, it means they are going to inhibit growth of bacteria. Then for the source, for the sources of the antibiotics, first we have the natural source. We have the semi-synthetic, and we also have the synthetic. For the natural source, these ones, you see that for them, they are naturally formed from microorganisms. These ones are naturally formed from microorganisms. For example, the it is streptomycin. Then for the semi-synthetic, these ones, they are natural. For them, the semi-synthetic, they are formed in the lab, but they are modifying the natural sources. And one of the main examples here is amoxicillin. While the synthetic, these ones are naturally formed in the lab. For example, the ciprofloxacin. So for the natural, they are naturally obtained from microorganisms. Then for the semi-synthetic, you're going to see that we are modifying the natural source. We are modifying these drugs that were obtained from the natural source. Then for the synthetic, for them, they are naturally manufactured in the lab. Lastly, we are going to look at mechanism of action. How do these drugs act? Remember, I had talked about the structure of a bacteria. Still here, you have to know the structure of the bacteria to know how the drugs act. For example, let's do the structure of the bacteria. We are having the capsule. This is our capsule. We are having the cell wall. We are having the cell membrane. But remember, on our cell wall, we are having structures like the pili. They're going to be all over the cell wall, and these ones are going to allow the bacteria to attach itself on different surfaces. Then the cell wall also has a flagella. It also has a flagellum, and this is used for movement. Then the bacteria also have cytoplasm within. We have what? We have cytoplasm. Within this cytoplasm, there are ribosomes. 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 Then there is a nucleoid. And we also have something called a plasmid. This nucleoid is the one that is having chromosome, DNA. This one is having circular DNA. This one is also the one that is having virulency factors. Virulency factors, these are substances which the bacteria use to infect their host, but also protect it from other substances. They protect it from the host, but they use it to infect the host. So we are still on our mechanism of action of antibiotics. Now these antibiotics, they are going to act on the cell wall and inhibit its synthesis. So there are antibiotics that are going to inhibit cell wall synthesis. Then there are also antibiotics that are going to act on the cell membrane. Antibiotics that are going to act on the cell membrane. How? They are going to disrupt. So they will be called. They are going to disrupt the cell membrane. So there are drugs that inhibit the cell wall synthesis. There are drugs that disrupt the cell membrane. Then we are also going to have drugs. We are also going to have drugs that act here on the ribosome. What is the function of the ribosome? Ribosome is one that is responsible for protein synthesis. So there are drugs that are going to inhibit protein synthesis. Inhibit protein synthesis. Also, there are drugs that are going to inhibit nucleic acid synthesis. And there are also drugs that are going to inhibit metabolic processes. Metabolic processes. Yeah, so those are the mechanism of action of antibiotics on bacteria. I think you might be asking yourself, what is metabolic processes? Metabolic processes, we see that in these processes, the cell is synthesizing what? The cell of the bacteria is synthesizing folic acid, and it is this folic acid that is very essential for the formation of what? Of DNA. Then nucleic acids, these are also formed in the what? In the DNAs. Yeah, so that's it for today's lecture. Thank you for watching. Don't forget to like and subscribe. I remain Helen Patience. Till next time, don't forget to log in into the next lecture. In the next lecture, we're going to be discussing the mechanism of action of antibiotics. We shall look at the cell wall, the cell membrane, and also look at the drugs, their side effects, and their indications. Thank you for watching.